Thank you for clicking on my video, and if you watch this video you'll have an explanation of my current hypothesis as to how most degenerative diseases are caused by long genes stopping working, and understand how that feeds into stem cell differentiation and gene expression, and have things to talk about with your healthcare professional which may be dangerous but might also improve your health. It's important to note I'm not medically qualified. I have a degree in physics but not medicine. I have been a member of parliament and I am a tech entrepreneur. I'm also a musician, but I'm not a doctor and this is not medical advice. I've studied these issues to improve my own health, particularly sleep. And I produced this video to be helpful to others with my research. My research is continually under review and I may not be right, but of course I think I am, otherwise I wouldn't produce it. My channel is not monetized and I'm not sponsored, but I do have 2 patent applications in. Please discuss the information in your video with your healthcare professional, and some things can cause harm for some people, so check with your doctor. If you don't have the time to watch the rest of the video, it's important to note that because of a shortage of a molecule called acetyl-CoA in the nucleus of some cells, longer genes producing longer proteins gradually stop working at the same frequency. The cells then don't function properly. And the effect of this is generally certain things don't stop working in the body or don't work as well. And it's possible to mitigate this, but it should only be done in conjunction with advice from a healthcare professional. Let's understand a little bit, go over the issue of DNA and producing proteins from DNA. So people, other creatures, plants, they will have lots of cells in their bodies, in their structure. A lot of the cells have a nucleus, not all of them do, red blood cells don't. And in the nucleus there is DNA, and the DNA is the same across the same creature. The DNA is controlled by a thing called the histone, and this opens and closes the DNA so that messenger RNA can be copied from the DNA. And the messenger RNA is used to build proteins. The process of opening the DNA is called acetylation of the histone, and closing it afterwards is called deacetylation. Genes vary in their length, and often a gene is so long that a number of transcripts are produced from it. If a gene has multiple transcripts, then the process called splicing happens to manage how the transcripts are produced. Sometimes not the whole gene is used to produce a transcript, and this can result in an alternate splicing, but that is not necessarily bad. For a transcript to be produced, the histone needs to be opened up or acetylated, and if not all the sections of a gene are acetylated, then not all of them will be produced. The process of acetylation involves a chemical acetyl-CoA, which is used to add an acetyl group to a lysine in the histone, and this is normally done by an enzyme either called HAT or CAT. Sometimes it's called CAT to emphasize its acetylating lysine. It might seem a bit odd being as lysine starts with the letter L, but lysine is an amino acid for which the letter K is used because leucine has L. Anyway, acetylation can occur without an enzyme, but that runs the risk of the body acetylating the wrong histone, doing very strange things. mRNA is created by a complex around an enzyme called RNA polymerase II, sometimes written down as RNAPII or RNA pol II. When this gets to a section of the gene, it introduces the acetylating enzyme. Without acetyl-CoA, the acetylation cannot happen and the RNA pol II's complex will stall. And if it stalls long enough, it will be terminated and the protein will not be produced. In 2022, a number of research papers were produced that confirm the proteins that are missing when cells don't work are the ones from longer genes. And you'll see links for these in the comments section. And also I've done a blog page about these, linking to all sorts of research papers. There have been also papers showing that as people age, they also encounter many alternative splicing, and splicing factors are produced that try to fix this but fail. What appears to be the case is that genes with 10,000 or more base pairs rather than proteins gradually stop working. And there are 3 theories as the cause of this. One is DNA damage, causing RNA pol II stalling. Now that's the theory the people who wrote the RNA pol II stalling paper. Another is from Professor Laurie Harris of Exeter University, that splicing factors are missing, and mine is that there's a shortage of acetyl-CoA which causes RNA pol II to stall. And there are 2 reasons potentially for lower acetyl-CoA. Cellular metabolism is quite complicated, and acetyl-CoA can come from a number of places, but a lot of it comes from the mitochondria as a result of the Krebs or TCA cycle. And a protein called SLC25A1 moves tricarboxylic acid from the mitochondria into the cytosol, and this is then used for various things, including creating acetyl-CoA. Sometimes there is too little SLC25A1, and sometimes the mitochondria don't produce enough tricarboxylic acid. Now, The reason why you get too little SLC25A1 can be because it's encoded by nuclear factor kappa B, and the Janus kinase at the edge of the cell reduces the proportion of NF kappa B as a result of cytokine interleukin 10 hitting it. And senescent cells produce SASP, which includes IL-10, and a stem cell with too little acetyl-CoA turns senescent because it can't properly differentiate. And a university thesis, which was not published in a journal, showed that IL-10 makes stem cells senescent. So the negative reinforcing feedback, where the more IL-10 there is in a body, the more senescent cells you get, and that's why I would think that you get patches of senescence in a body, that is, patches of the senescent cells that are not properly functioning. They don't properly differentiate. Alternatively, you can have too little tricarboxylic acid. The mitochondria turn glucose into ATP, and they also generate TCA via the non-canonical TCA cycle, which involves SLC25A1, of course. And over time, mitochondria deteriorate for various reasons. There is a process called autophagy, which chews up bad mitochondria and other organelles and replace them with better ones. And autophagy depends on a long gene called Becklin-1 and possibly other long genes, and hence reduces over time because long genes reduce. And this is a second negative feedback system that operates through acetyl-CoA, but it operates in an individual cell rather than operating throughout the body, but it still has the same effects in the sense it reduces the probability of long proteins being produced. So I did an experimental cutting of the Gordian knot to see if this would work, and I worked out some proposals for basically increasing acetyl-CoA in the nucleus and doing that to myself and a few other people. And apart from studying what happens with my hair and functional tests, I've also done weekly blood tests, and I've seen improvements in a number of biomarkers as well as improvements with certain functional tests, and that's in a video I produced in December, which is on the same channel. At the moment I want to be careful to make sure that any further people experimenting with the same protocol are well informed and with proper monitoring by a healthcare professional. So it will publish late in 2023, but it's not being published at the moment. So my company offers at no charge an educational program on how to experiment with this protocol, but it's not health advice or medical treatment. If people wish to study it, that is, if students actually wish to try it out on an experimental basis, students agree to have frequent blood tests, not necessarily weekly, but there is a cost to this that the students have to pay for. Students will have to purchase their own molecules. It involves consuming various molecules, which are generally available over the counter in most, if not all, countries. A few may require a prescription, but they're not essential. Things like rapamycin. You don't necessarily have to have rapamycin. Students must sign both a non-disclosure agreement and a disclaimer, and the students stay in touch with me and take advice from their healthcare professional to ensure no harm is done. Please see the comments section for the email address to use to apply to join the education process, which is coached biohacking, and it's at your own risk. So thank you for watching the video.